Well, I want to express how honored I am to be here, to be contributing to this very prestigious series of lectures, and hope that I can induce you to put many zeros after any number you put onto those forms that David referred you to. The topic for today is this one, human twinning and human individuality. Twins have long been the subject of myth and mystery in the human imagination. Here shown Romulus and Remus, the legendary founders of Rome, as perhaps our most famous examples of twins. But twinning didn't become philosophically or politically controversial until this event, the birth of Louise Brown by the very first test tube baby in 1978. The development of in vitro fertilization raised considerable ethical concern at the time regarding the use of embryos for this procedure. And in support of this practice, a rather prominent biologist in the United States, Clifford Grobstein, sprung to the defense of IVF by proposing the concept of the pre-embryo. So here in a paper published in 1979 in Scientific American, Grobstein defends IVF, stating, "The stages involved in IVF not only are pre-persons, but are also pre-embryos. They are pre-embryos because separated cells from the early cleavage stages of the eggs of non-human mammals, and most likely of human eggs, can each give rise to a whole embryo in twinning. And because cells can be deleted from early clusters, or two clusters can be combined, and a single embryo can still result. Therefore, the cellular stages prior to implantation, a single embryo has not yet been established." So this argument was, of course, taken up by many other authors and expanded upon and reiterated over the years. But where has this argument ultimately led? The view that no human individual can exist so long as twinning is possible has led to the denial of the humanity of early human embryos. The view that an embryo is merely a cell on the way to becoming a human being, but not there yet, a pre-embryo, for example, affords embryos little or no moral value. And many countries have established research policies that allow experimentation on human embryos prior to individuation, when twinning is no longer possible, that is, after the 14th day of life. For example, in this country, the report of the Warnock Committee in 1984 very much endorsed the analysis of Grobstein, stating at the start of cleavage in the 2 or 4 cell embryo, each cell retains its totipotential capacity. Thus, if separation occurs at the 2 cell stage, each may develop to form a separate embryo, and such a separation could lead to identical twins. A human embryo cannot be thought of as a person, or even as a potential person. It is simply a collection of cells. So the question of twinning, and whether or not any of us was ever a one-celled embryo or zygote, was also taken up by Anscombe, who I believe actually contextualized this question in the most intelligent and critical manner, addressing in this essay, Were you a zygote? She addresses the possibility that identical twinning involves the splitting of a pre-existing human, similar to the division of an amoeba in mitosis, and states, "The obvious objection is that in the case of an amoeba, there is no doubt that we start with one amoeba, and it splits into 2 amoebas. But in the human case, it precisely is the question whether what we start off with is a human." So this is why twinning is a problem, and I'm going to spend a little bit of time hammering this insight and making sure that it's completely clear, because it's going to become the basis of much of the remaining analysis I'll be doing. Anscombe's insight is that the timing of twinning really matters. If we start with an entity that clearly manifests the characteristics of a human person, it's conscious, rational, can converse with us, twinning after the nature of an entity as a human being has been clearly established does not call the ontological status of the original human into question. So in this case, if we could, for example, twin or clone this individual, perhaps by removing some skin cells from their hand and undergoing a process of cloning to produce a little miniature identical replica of this mature human, it would not call the identity of this human as a human into question, would it? Although we've produced an identical twin through this procedure of cloning, we don't have any question about the fact that what we started with was a human because it was already established as a human. In contrast, what about one of these? So this is a one-celled human embryo shortly after its formation. And if that cell divides to produce two cells, and each of those cells goes on to produce two mature human individuals, it does call the question of that original cell into question. Was it a human person who divided into two human persons? Or was it perhaps just a human cell that produced another copy of itself? And at some point later, those cells produced a human person. So put another way, the problem of twinning looks like this. If the zygote divides to produce two identical totipotent cells, both of which can, if separated, develop into a mature individual, how then can the original zygote be considered a single human person? So to address this problem of twinning, I'm going to take two separate tactics. I'm going to start, because I'm a biologist, with the biological issues and cover the following questions. When does a zygote form, and what actually happens in the early stages of human development? How long does totipotency, or the ability to independently generate a full human being, persist? When does twinning actually occur? And then we're going to move on to the philosophical issues. We're going to address Anscombe's concern. Is there any evidence that the zygote is an actual human being rather than merely a human cell? And in the remaining time, cover the remaining concerns raised by twinning. So fortunately for us, the question of what is the zygote and when does it arise answering that question is facilitated by the fact that science actually has some pretty clear criteria for the formation of new cells. Changes in cell type, either in the laboratory or in the embryo, are detected by only two criteria. Either a change in material composition, so what a cell is made out of, most typically a change in gene utilization, or by a change in cell behavior. And often these two things come together. If you change what a cell is made out of, change its molecular toolkit, you can change how it acts. These criteria are used throughout the scientific enterprise to determine when a new cell has formed, either in the laboratory or in an embryo. And of course, if a new cell arises from two or more pre-existing cells, logic requires us to conclude that those cells have to cease to exist before we can get a new cell. So based on these criteria, is the product of sperm-egg fusion a new cell, or is it just a modified gamete? You often hear the product of sperm-egg fusion referred to as a fertilized egg. I hope to convince you that that's actually a horrible description, and in fact not a scientific one at all, and that the term fertilized egg refers to nothing. There is no such entity. But in contrast, a new cell unambiguously forms at sperm-egg fusion. So this is an electron micrograph of a larger egg cell and a sperm cell fusing to each other. And upon sperm-egg binding, or the attachment of these two cells to each other, the plasma membranes, the outer surfaces of these cells, will fuse to each other in a very well-studied and rapid event that takes less than 250 milliseconds to complete. So there is an actual instant where we go from two cells to one cell. And once that fusion has occurred, all of the lipid and protein that used to be uniquely present on the surface of the sperm is now free to diffuse into the surface of this new cell, changing its molecular composition. In this process, the sperm will rupture, ejecting its nucleus and its cellular contents. And clearly the sperm as a separate cell ceases to exist in this instant of sperm-egg fusion. What about the egg cell? Well, upon fusion and destruction of the sperm, the resulting cell has a new molecular composition that's distinct from that of an egg. We've already talked about the incorporation of sperm-derived lipid and protein into the surface, but the changes are way more extensive than that. This cell now has a new genetic composition, with half of its DNA derived from mom and half from dad. But more importantly, this new cell also radically changes its behavior and acts in a very un-egg-like manner. So the entire raison d'etre of being an egg is to find a sperm and to fuse to it. That's how the egg is designed. And yet, within 1 to 3 seconds following that instant of sperm-egg fusion, this new cell will initiate a molecular cascade that ultimately, over the next 30 minutes, will result in the release of enzymes into the space surrounding the cell. And the function of those enzymes is to destroy any remaining sperm binding sites. So the entire function of the egg is to bind and fuse to a sperm. The very first action of the new cell that's produced is to antagonize the function of the gamete from which it was derived. So the egg, as a haploid gamete designed to fuse to a sperm, clearly also ceases to exist in this instant of sperm-egg fusion, and a new cell with unique material composition and behavior is formed in this event. I'm gratified to say that Anscombe also agrees with this conclusion. In answer to the question of whether I was once a sperm and an ovum in this same essay, she replies, no, because the objection to this is just that the sperm and the ovum were not one substance. Whether the zygote is a new human substance, that is, a new human being, is Anscombe's central question that we will return to once we've dealt with the simpler and perhaps more trite biological questions. So once we've generated that new cell, the zygote, what happens? I'm going to show you a movie of the first 5 days, roughly, of human development taken from a fertility clinic that's listed here. What you're going to see in this movie, the central sort of grainy gray cell is that one-celled human embryo or zygote. It's surrounded by a jelly-like coat that does not contain any cellular material, known as the zona pellucida, or the clear zone, that protects the egg and the early embryo during these first 5 days. And the larger, brighter circle is the culture well that the embryo is being maintained in. So what you can see in this movie is that the egg enters into the embryo enters into a rapid period of cell division, generating 2 cells at about 24 hours and 4 cells at the end of the second day. These early cells are known as blastomeres. And by the 3rd round of cell division, the embryo reaches what's called the morula stage. And the cells of the morula are going to change their molecular characteristics to adhere more tightly to each other in a process known as compaction that you can see here. Once the cells are really tightly stuck to each other, that allows the embryo to transport fluid to the inside and blow up, kind of like a fluid-filled soccer ball, to reach the blastocyst stage that's shown here. So the blastocyst is important because it's the first point in human development where we have 2 distinct cell types that are going to contribute to different things to the formation of the mature body. The outer cells of the blastocyst, those sort of black and white hexagons of the soccer ball, are known as the trophectoderm, and they're going to do 2 really important things. They're going to mediate implantation, so they're going to attach to the uterus and allow the embryo to implant, and they're also going to produce the structures of the placenta. A small group of about 8 to 10 cells on the inside, creatively named the inner cell mass, or ICM, are going to produce all of the tissues that you carry around in your body right now. All of these events we've just seen in the movie take place in the woman's reproductive tract. So this is a cartoon of an ovary with a maturing egg cell that is ovulated out into the fallopian tubes, which is where fertilization typically occurs, and the formation of the morula and the blastocyst, with implantation happening at about 6 or 7 days. After implantation, a couple of important things happen. These inner cell mass cells divide into 2 layers, an upper epiblast and a lower hypoblast. And this little space up above is the beginning of the formation of the amniotic cavity. At about 14 days, an important event occurs called gastrulation, or the formation of the primitive streak. So in this event, cells from this epiblast are going to dive down deeper in the embryo and turn that 2-layered embryo into a 3-layered embryo. This is also the very first point at which the earliest stages of neural development commence. So, primitive streak formation or gastrulation. So that's what happens in early development and when we first get a zygote. But it doesn't really address the question of how long totipotency persists. So as you will recall, when we were thinking about the problem of twinning, the first concern regarding twinning is whether the zygote simply divides to produce two cells, as does an amoeba, and each of those totipotent cells can now go on to produce their own independent individual. So, what is totipotency? Totipotency is the ability of an isolated blastomere to independently generate a complete developmental sequence, resulting in the formation of a mature individual. The zygote is clearly totipotent, but how long this potency or this property will persist in the progeny of the zygote is actually a really unclear question, a poorly studied question. I think the two best papers addressing the question of how long totipotency persists were actually only published within the last couple of years, both by the same group. One of which, published in 2017, clearly demonstrates that the ability to independently generate your own independent human, or in this case a mouse, will segregate. That capability segregates between the first two blastomeres, in most cases with only one cell remaining totipotent after that division. And the second paper, just published a couple of months ago in September of this year, reveals that that difference in the developmental ability of the first two cells reflects the inheritance of different components from the egg. So a lot of factors that are known to be important for normal development, for example, this one, COX-3, are enriched in one side of the mature egg cell. So if that egg is fertilized to generate a zygote, and that zygote goes on to divide into two cells along this plane, what you end up with are two different cells that have different amounts of this factor present in them. If you separate those cells, they'll go on to generate independent blastocysts, but the blastocysts will not have the same properties. So here, blastocysts or sister blastocysts derived from each of these first two cells are stained with to reveal the presence of three factors that are known to be required for embryonic development. This one, that had lots of COX-3, has good, strong staining of all three of these colors, green, red, and blue. But this one is only very weakly stained for two of these critical factors, suggesting that it's not going to be very good at going forward in development. And in fact, that's exactly what they saw. They saw live births in only 16% of cases where these two blastocysts were both transferred to the same surrogate mom, and twins in only 6% of the transfers. Put a different way, 84% in 84% of embryos, neither of the first two blastomeres remains totipotent. Does that make sense to everyone? Good. So that conclusion is actually supported by a very large body of data looking at differences in the properties of blastomeres in the mouse model. So for most embryological studies, the vast majority of them are done in mouse because mice are very readily available, they're easy to study, and we have a lot more power to manipulate them than we do to manipulate human embryos, thankfully. But I'll show you just one example. This is a case of a 4-celled embryo looking at the nuclei of each of those cells, and then revealing the presence of a particular protein that binds to DNA within the nucleus. And the important take-home here is all 4 of those circles look about the same. But that's not the case for all factors that interact with DNA. In this case, you can see one cell has an awful lot of this particular protein, and some cells have almost none. If you correlate the distribution of that protein with the function of the nucleus, so here in red is shown the amount of this protein, revealed here in the picture, and in green is shown a measure of the activity of the DNA. You can see those two things are very highly correlated. When you have low amounts of protein, the nucleus is really quiet. It's not doing much work. And when you have high amounts of protein, you have high amounts of activity in the nucleus. So that tells us that at the 4-cell stage, cells have different properties and they have different functions. Do those functions actually translate into different developmental abilities? And the answer to that is also yes. And we know that from experiments like this. If we take 3 embryos that are all dividing in the same pattern, and we isolate 1 red cell from each, 1 blue cell from each, and 1 green cell, and then aggregate those cells together and transfer them to a surrogate mom, what we find is in 85% of the cases, the red cells can produce a live-born pup, but in none of the cases can the green cells do it all on their own. And what that tells us is that at the 4-cell stage, blastomeres have distinct patterns of gene expression, they have different functional properties, and they have different developmental properties. Put in other words, the full developmental potency is not possessed by any one cell at this stage. So what do we conclude about totipotency? In 84% of the cases, neither of the first 2 blastomeres is totipotent. Only 6% of the cases are both of them. There is considerable variability among species, but it's likely that in most mammals, totipotency does not persist beyond the 2-cell stage. A number of factors known to be important for development are differentially distributed in oocytes and also in the first 2 cells. And clear differences in gene expression and developmental potency are exhibited by the 4-cell stage. That tells us blastomers are not equivalent. Okay, so in light of this restriction of totipotency, when does twinning actually happen? When do we get identical twins? Which brings us to sort of our second concern about twinning, which is whether, in the rare cases where 2 totipotent blastomeres actually ever exist, do they ever independently generate identical individuals? I.e., do they ever actually produce twins? So I'm going to emphasize that monozygotic twinning is actually quite rare. It occurs in only about 0.35%, less than 1% of all births. In assisted reproductive technologies, the incidence of monozygotic twinning is actually quite elevated to about 2% of all live births. And the timing of twinning is historically believed to be inferred from the arrangement of the embryonic membranes. So bear with me, it's a little complicated, but not too bad. So let's focus on this first column A. So twinning before the formation of blastocyst, our little soccer ball here, either by separation of the first 2 cells or a small number of cells before blastocyst formation, would give us 2 independent blastocysts, each of which is going to make its own amniotic cavity and its own surrounding chorionic cavity. So these are so-called dichorionic, diamniotic twins. If we focus on B, if we twin after the formation of the blastocyst by separation of that inner cell mass into 2 groups, each of those little groups is going to make its own amniotic cavity, but they're all going to be inside of 1 chorionic cavity. So these are now monochorionic, diamniotic twins. And that's about 66% of all twins. If you twin after the formation of the amnion, this is a very rare event, less than 1%, and we get either conjoined twins or so-called monochorionic, monoamniotic twins contained within a single amniotic cavity. So that's what we think happens. Our question is, does this ever happen? Do we ever see twinning really early at that 2-cell stage, reflecting totipotent blastomeres? So substantial evidence, as we've just reviewed, suggests totipotency does not persist. Besides, despite a very high incidence of monozygotic twinning in IVF and routine daily observation of IVF embryos, it's never been reported. With one author saying, we have never observed an embryo splitting in half before the blastocyst stage on its own initiative in over 15 years of laboratory experience. And lastly, early blastomers are very sticky and they're very strongly inclined to act as parts of a single embryo. We know that from a number of experiments. If we dissociate early embryos into single-cell suspensions and let them reaggregate, they sort back out, reestablish their same initial relationships, and go on with development. Even if you take 2 embryos and aggregate their early cells together, you make a chimeric embryo that contains more than 1 type of cell. Here, in fact, is a mouse that's comprised of 4 different types of stem cells, all of which were colored a different color so we could trace where they are. But you can even make chimeras out of different species. These were geeps that were produced by fusion of a goat embryo and a sheep embryo, which would produce an animal that had half of the cells of each of those 2 animals. So the idea that you could actually have two embryos developing from the first two blastomeres has been directly tested by this experiment. If you produce a single zone of pellucida that has two mature egg cells in it and fertilize each of them, you get fraternal twins. Different sperm, same egg nucleus, same egg genome. If you watch what happens to those fraternal twins, at the Memorial stage, when compaction occurs, they form a single blastocyst that is comprised of two different genomes, a chimeric blastocyst, and they continue on in development as a single entity. So even if, through some magical means, we could do this, separate the blastomeres at the first two-cell stage, they would likely to refuse into a single individual. So there's good evidence that in most cases, the first two cells are not totipotent. There's no known mechanism for separating those cells, and it has never been observed to actually occur. Consequently, twinning at the two-cell stage is highly unlikely. So how then do we get this 33% of twins that have two chorions and two amnions? I would say it happens at hatching. You remember I told you that the cells of the trophectoderm are responsible for implantation, but they're enclosed within that protein coat, and they have to escape. I'm going to show you a movie of how that happens. Here, the blastocyst grows bigger and bigger, importing more and more water to the inside, and it also secretes enzymes to break a hole in the zona, and then the whole embryo squeezes out. So I would propose that this is a point where monozygotic twinning could occur. And in fact, this has been observed in IVF clinics. I'll show you just one example. So here's an embryo hatching from the zone of pellucida. And it actually generated two blastocysts, each with a subset of the ICM cells inside. When these three embryos, so this would be a fraternal twin here and an identical twin here, were transferred, they resulted in a triplet pregnancy. Only one fetus survived, which indicates that we can have identical twinning at the blastocyst stage. And I would conclude that this never occurs for all the reasons I've just described, and that all twinning is likely to occur after blastocyst formation. So, are we done yet? Or do we have any credible concerns still remaining about twinning? The biologic facts make it extremely unlikely that twinning ever occurs at the two-cell stage. However, even if it doesn't, even if it's an extraordinarily rare event, I think the philosophical questions still remain. And the most significant concern is the one raised by Anscombe, who asks whether there's any evidence that the zygote is actually a human being. So, what is produced by sperm-egg fusion? Its scientific name is the zygote, or one-celled embryo. But the challenge is looking at this cell doesn't tell us much about its nature. Is this a new individual human being, or just perhaps a new kind of human cell, a pre-embryo? And at early stages, recognizable human form and function are not evident. So how do we know that a zygote or a morula or a blastocyst is a human being and not just a cell or a ball of cells? This is a surprisingly difficult question to answer. But the answer turns on this question: what is an organism? So Merriam-Webster gives us a couple of definitions. A complex structure of interdependent and subordinate elements whose relations and properties are largely determined by their function in the whole, or an individual, organized, constituted to carry on the activities of life by means of organs separate in function but mutually dependent, a living being. And I would note that a living being of the genus Homo sapiens would be a living human being. So both of these definitions stress the essential feature of an organism, which is that it's a complex thing, it has lots of parts, and those parts don't work together for the good of the thing as a whole. The challenge in distinguishing a multicellular entity like us from a single human cell is that cells are also clearly organisms. I'm going to show you a very short movie of a cellular organism. This is a human neutrophil in culture. This was a movie made in 1958 at Vanderbilt University. The star is a human neutrophil whose job in the body is to search out bacteria and destroy them. These little round or cellular-shaped cells are blood cells, and then the little dots are bacteria. And what the movie illustrates is that cells exhibit autonomously coordinated activity in parts for the good of the cell as a whole. Or, in other words, cells are organisms. So here you can see this cell is capable of movement, which requires the coordination of lots of intracellular components. It's clearly capable of detecting its environment. It knows where that bacteria is. It can avoid obstacles, reorient as it chases its prey, and eventually gets it. Yes. The suspense always kills me in this movie. So what this shows us is that cell type specific properties and functions persist ex vivo, and activities that can be explained by the properties of cells are not sufficient to conclude to the presence of a human being. Which brings us to the central problem thinking about twinning, as well as about the beginning and end of human life. Both human cells and human beings are alive, but human beings are not just a collection of living cells, so how do we know the difference? One of the concepts that comes to our rescue here is multicellular versus unicellular organisms. Multicellular organisms, such as human beings, are uniquely capable of development, the self-directed maturation to a characteristic adult form. Adaptation and wound healing exist to preserve the healthy function of the multicellular whole rather than the individual cellular parts. And interactions between the cellular and subcellular parts are ordered to the good of the multicellular whole. So development, adaptation, and integration will distinguish human cells from human beings. So let's go through those one at a time. Well, growth towards a characteristic mature form is the entire business of what an embryo does. This is the first 8 weeks of human development, actual human embryos taken from the Carnegie collection, with our zygote being a little dot here above the number 1. And in a short 8 weeks, it generates a full and complete, albeit very small, this is 5 millimeters, human form. What about adaptation? I'll give you just one example. Pre-implantation genetic diagnosis. So here an embryo is immobilized by suction against a pipette with another pipette inserted. A single cell in this procedure is typically removed from an 8-cell embryo when blastomeres are likely to have specialized developmental functions. Such biopsied embryos are allowed to develop to the blastocyst stage, and if the tested cell turns out to be genetically normal, then it's transferred to the uterus. And remarkably, embryos are able to recover from what can only be described as a catastrophic injury. You know, to lose 1/8 of your body mass at such a tender age is a pretty big hit. And yet, embryos will regenerate the missing tissue, such that there have been many thousands of live births from such biopsied embryos worldwide. A review that I recently wrote in the journal Law and Public Policy in 2014 goes exhaustively through the scientific literature and identifies dozens of early and ongoing integrated events taking place minutes, hours, and days after sperm-egg fusion. It's absolutely clear from this evidence that sperm-egg fusion sets off a molecular cascade that controls events that take place hours or even weeks later. These events are not required for cellular life. And in fact, they're only intelligible as part of a developmental sequence that's produced by an organism. In fact, that very first event I mentioned to you, the release of enzymes. Why does a cell want to do this? Well, to prevent more than one sperm from binding, which would increase the amount of DNA present in the cell. And yet, cells are perfectly happy to have dozens of nuclei. All muscle cells in your body have many, many nuclei, and they function perfectly fine. So a cell doesn't care how many nuclei are present. The only thing that cares is an embryo. And an embryo cares because the nature of development requires there to be a balance between the maternal and paternal pattern of DNA organization. And so having 2 sperm is going to offset that balance and destroy development while doing nothing to the function of the cell. And while some scientists may dislike and or deny this conclusion, it's supported by hundreds of peer-reviewed papers, and it's entirely uncontested in the literature. So I'm not just cherry-picking the papers I like. There are no papers that say anything different than this. What you will find are papers where scientists will assert that nobody knows or that their personal opinion is, but I have defied everyone I know to send me a single shred of evidence, and in more than 10 years have received none. So what can we conclude about the nature of the zygote and the implications for twinning? Well, a new cell, the zygote with unique material composition and behavior, comes into existence in this instant of sperm-egg fusion. It exhibits all the characteristics of a full and complete, albeit immature, human being. Therefore, human life commences with the onset of organismal function at sperm-egg fusion, or a human zygote is a human being. And this is an objective, evidence-based definition of when human life begins. Its implications for twinning are this, that whether twinning occurs at the two-cell stage or later, it disrupts the ongoing developmental sequence of a single organism, and therefore does not call the nature of the original embryonic human being into question. All right, so now for the last little bit, we're going to go through the remaining questions raised by twinning. I'm just listing some of the works I discuss in this section. A very seminal paper by Joseph Dunseel, Norman Ford's book, When Did I Begin?, papers by Wallace, Regard, and of course Anscombe, and Saul Kripke, who I think provides us a critical insight in this general area. I'm also going to refer you to my book that's coming out. David asked for the cover photo, which looks like this. In chapter 5, I go over a number of philosophical concerns, but I'm going to focus on just a few today. Virtual humans, or the different meanings of potency. Does the original embryo die, or perhaps persist? Who are the parents of the twins? And identity in form and matter versus ontological identity. So let's start with that first one. So Dunseel and Ford are the ones who most clearly raise this issue, with Dunseel saying that if potential to form a human is the criteria for being a human now, therefore before the loss of totipotency, every single cell of the zygote, morula, and blastocyst is a human person. Ford agrees and attempts to resolve Dunseel's problem by holding that while a zygote has a natural active potential to become a mature person, it's an individual, not a person. And thus, twinning replaces the non-human individual with two human persons. So again, we need some modifications of this argument in light of the facts we've just reviewed. Since we know totipotency doesn't persist likely beyond the two-cell stage, therefore, in most cases, only one developmental trajectory is initiated, and in the worst case, only two. It's never the case that we have every single cell of the embryo being a human person. Both when twinning occurs at the two-cell stage or later, a single organism is split. So Dunseel and Ford would presumably have to maintain ensoulment is therefore delayed for all embryos, but this leaves us in the awkward position of claiming that a human entity that's clearly a human organism and undergoing development is somehow not a human being. I think the central issue in this reasoning is the question of potency. If a zygote is a person in virtue of the fact that it has the potential to develop into a person, then all cells with this potential are equally persons. A zygote that has the potential to be two persons cannot simultaneously be a single person. And I think we can resolve this by having a better understanding of the word potency and what it really means. So Aristotle considers potency along two axes, active and passive, proximate and remote, with active potency being the power to act in a specific manner that is intrinsic to the thing itself. So I have the active potency to speech even if I'm currently silent. Passive potency, on the other hand, is the power to be acted upon in a specific manner. So wood has the potency, the passive potency, to be fashioned into a chair by a carpenter. Water does not, and neither of them has the active potency to become a chair on their own. So let's consider proximate and remote. Proximate potency is the power to either act or to be acted upon in a specific manner that can be immediately exercised. So I have the proximate potency to speech even if I'm currently silent, which no one has yet ever experienced except when I'm asleep. Remote potency is the power to act or to be acted upon in a specific manner that requires some intervening event before it can be exercised. So an infant has the remote potency to speak, but it has to learn how first. A sapling has the remote potency to be fashioned into a chair, but it has to grow into a tree first. So what are the kinds of potencies we see in development? The zygote exhibits a proximate, active potency to be a human being. The zygote is a human now because it is actively exercising an intrinsic power of maturation or development, such that in an appropriate environment of its own authority, it's going to produce one of these. It's going to produce out of itself a mature state. In contrast, early blastomeres have only a remote active potency to be a human being. So blastomeres are parts of a single human now, but they can become an independent human if an intervening event occurs, which is the isolation of the blastomere. So the remote potency of blastomeres means that there's a single, unified, individual part of the twinning, and that blastomeres must cease to be a part to actualize their remote potential to become a whole embryo on their own. In other words, they must undergo a substantial change. Coming back to Dunseal and Ford, proximate active potential to develop into the mature human is the criteria for being a human now. This potential exists in a zygote, but not in totipotent blastomeres, even when they actually do exist. And upon splitting, the remote active potential of blastomeres or parts to independently develop as a full human becomes proximate. And at that point, two humans exist. So does the original individual die or persist? This is an objection raised by Ford and Anscombe, as well as others, the no corpse objection, with Ford saying, it is impossible to admit that the original zygote in human individuals cease to exist when, without dying, and without a dead cell remaining, it gives asexual origin to identical twin offspring. Anscombe echoes this concern, stating, but what has become of the human that both of them, the twins, were once identical with? Has he or it simply ceased to exist, as we might say the parent amoeba ceases to exist on splitting? Well, I think Anscombe is following the right direction. In many events, something new is produced without a corpse being required. In mitosis, for example, two cells are produced out of a single cell without anyone dying or leaving a corpse behind, as you can see here quite elegantly. Similarly, sperm and egg will fuse to produce a zygote without the death of either gamete or the production of a corpse. So substantial change does not require a death. It really requires a ceasing to be. Apt matter can persist and receive a new form, yet there must be evidence for a new form, a change in composition or behavior. So is there any evidence that the embryo ceases to be after splitting? I would say no. And I would say no based on an experiment like this. So again, using mouse embryos, they were split at the late morula stage. The demi-embryos and the intact sibling embryos were cultured in parallel, and we're going to see what happens. So this first column is the half or split embryos, and these are the whole embryos, the sister whole embryos. And this is what you see. So they begin forming a blastocyst at exactly the same time, and expand at exactly the same rate, with the only difference being the half embryo has half as many cells. So demi-embryos do not restart from a totipotent state, but rather they regenerate and develop in synchrony with intact siblings. So what does this mean? I think if an embryo is split in half, and one half is destroyed, as we saw in the last movie, the surviving half immediately continues development as a unified whole. There's no evidence that the original embryo has ceased to exist. Therefore, it's reasonable to think of the surviving half that we started with an embryo, the surviving half as an injured embryo, and the entity that continues immediately through development is a healed embryo. And logically, the ontological status of this half does not depend on the as of yet undetermined status of the second discarded half. So what about the second half? If the embryo is split and both halves survive and regenerate, there's still no evidence that the initial embryo ceases to be, suggesting that one of the twins is ontologically identical to the original embryo and one is newly generated. Our inability to tell which twin is the original is an epistemological problem. It's a problem of knowledge, not a problem of ontology or being. So, who are the parents of the twins? This is an objection that was raised most clearly in my mind by Wallace, the unnatural reproduction question, or who's your daddy? Twinning would, quote, be an incidence of asexual generation, the production of one species from another species, a type of generation found in lower forms but not proper to humans. In light of everything we've talked about, is asexual reproduction natural to humans? It certainly didn't feel that way to Wallace, but I think the evidence clearly indicates that in the first 14 days of life, it simply is one of the ways we reproduce. Underlying Wallace's objection is the question of parenting and the relationship of the twins to each other. In Wallace's view, if twinning initiates with an actual human being, the zygote, then the original person or zygote is both the brother and the sister, or the father and the mother of the twin that's generated, and this appears to be unseemly. However, I believe that this concern can be addressed by drawing a distinction between random and intentional acts. So what is intention? Aristotle, or Aquinas, gives us this: the rational or natural ordering of an act to a specific end. And he says, in Summa Theologica, to intend is to tend towards something, which is indeed said of both the mover and the thing moved. Accordingly, that which is moved to an end by another is said to intend that end. In this way, nature is said to intend an end as it is moved to its end by God, just as the arrow is moved by the archer. And in this way, also brute animals intend an end as much as they are moved by natural instinct to something. Importantly, intention does not require choosing. The arrow intends to hit its mark. Swallows intend to return to Capistrano. Neither of them makes a rational choice to do so. Now, some acts, such as authorship and creation, can only be intentional. If I have a monkey sitting at a typewriter and he randomly bangs out the text of Shakespeare's Hamlet, the monkey cannot be legitimately named as the author of this work because the act of striking typewriter keys is not naturally or rationally ordered to any specific literary end. Similarly, if the growth of a tree randomly produces an image of the Virgin in the bark, the tree cannot be legitimately named as the artist creating this image because the act of growth is not naturally ordered to the production of religious images. Okay, here comes the tough one. Parenting is also intentional. The terms mother and father most properly refer to those agents who intend to initiate a sexual reproductive act as reproduction. Whether through natural instinct in animals or through conscious intention in humans, whether out of love of pleasure, out of love of each other, or even out of violence in the case of rape. Mother and father refer to those agents who act for the end of reproduction, who act intentionally. Twinning, in contrast, is an unintended outcome of an act that's naturally ordered to development, not reproduction. And consequently, if twinning occurs as an unintended consequence, due either to the zygote or to some external agent, it is the father and mother of the original zygote as the intentional originators of the reproductive act who are the parents of the twin. Okay, last one. I think I can do this in like 2 minutes. Identity informed in matter versus ontological identity. So many people who are concerned about twinning basically have this in the back of their heads. The transitive nature of identity requires that if A equals B and A equals C, then B must also equal C. And if the original zygote is identical to the two totipotent daughter cells, then both daughter cells are ontologically identical. So we need some modifications of this argument. First off, twinning is unlikely to occur at the two-cell stage, and substantial evidence that I did not review here today suggests that in most cases the first two blastomeres are not totipotent and therefore not identical, although we did talk about some of that later. Lots of data indicates that identical twins are epigenetically distinct even at very early stages, so consequently zygote A is unlikely to be identical to both of the daughter cells, although it may be nearly identical. So there are two related responses to the ontological identity argument. Identity or near identity is neither sameness nor ontological identity, and Kripke's insights on naming. So I'll just give you a few examples of near identity. Eggs are much bigger than sperm, such that after sperm-egg fusion, the resulting zygote is 99.9% molecularly identical to the egg that gave rise to it. And yet, a zygote is a person, and an egg is a cell, so they are not the same. By most measures of genome identical nature, this organism and this organism are 98% the same, and yet they are clearly not identical. And in the case of death, immediately prior to and following the time of death, the soul will leave the body, but there's no molecular change in the composition of the body. It's identical before and after, and yet a corpse and a living person are clearly distinct entities. So Kripke gives us a way out of this problem by providing an important insight into, by drawing a distinction between naming and identity. That the attributes by which we fix a name do not make the individual be what it is. In more precise terms, the substantial identity of a thing is logically prior to the attributes that distinguish that thing from anything else. And I'll give you Kripke's own example. We may know Benjamin Franklin to be Benjamin Franklin because he's the inventor of the bifocal. And we may know Thomas Jefferson to be Thomas Jefferson because he's the author of the Declaration of Independence. But what if we're wrong? What if it turns out Franklin actually did not invent bifocals and Jefferson did? This fact does not turn Jefferson into Franklin. An attribute used to name or distinguish one human from another does not make them distinct. So let's apply this to our presumed totipotent blastomeres. Even if blastomere A and B are identical, and there are no unique attributes distinguishing them, they are not made ontologically identical by this fact. It may be difficult or even impossible to fix a name, but in Ford's words, they have separate, concrete existence, and they are distinct ontological entities. So the difficulty of fixing a name is an epistemological problem, not an ontological problem, and I would note that it's one that is likely to persist to much more mature states of development. I personally would have a very hard time distinguishing between these two individuals. So twinning conclusions. Twinning is likely to occur between the 4 and 8 days of life. In most embryos, the first two blastomeres are unlikely to be identical and therefore not totipotent. Proximate active potential to develop into a mature human is what makes a zygote a human now. Totipotent blastomeres, even in cases where they do exist, have only remote active potential for development. Substantial change does not require a corpse. Asexual human reproduction is in fact entirely natural until day 14, although it may still be unseemly. Parenthood, or begetting a child, is an intentional act, and attributes used to fix a name do not confer ontological identity. Or, put other words, identity is not sameness. So with that, I'm going to stop, almost exactly at 50 minutes, and take a few questions. These are the two books I mentioned earlier, the book that I wrote with my brother Samuel, who's a philosopher, and the book that's coming out on this topic, and also the topic of chimeras, or the production of a single individual from two individuals, will be appearing in February of 2020. Thank you very much for your attention.